What are all the jobs? Only one single cell, like for example in unicellular it is yeast, on one single example. So everything the one cell is doing, right? So what are all the things they are doing, the one cell? For example, yes, respiration, excretion, reproduction. So these are all the things the only one cell is doing. So for our trillions of cells here, right? So in this chapter we are going to see about the cell. So in NCERT, so you are all having books, right? So plus one NCERT. So we are going to see in line by line. Okay. When you look around, you see both living and non-living things. You must have wondered and asked yourself, what is that makes an organism living, or what is it that an inanimate thing does not have which a living thing has? The answer is the presence of the basic unit of life. The cell is all living organisms. So we are all looking around in there. So we are an human beings, that we are an organisms. So like this chair, this pen and mouse and all, and in non-living things. So we are all living things. So we are going to see about our living things, but we need some cell to live. We want cell to live in a correct position, right? So here all organisms are composed of too many cells, or single cells or multicellular. So single cell, unicellular is a single cell organisms. So only one cell, for example yeast cell we have already seen. And then for multicellular organisms, so example human beings, right? So plant cells, fungi, plant cells and animal cells, human beings, these are all the multicellular organisms. So apart from yeast, give some example, only one example for a unicellular organism. Bacteria. So okay, and then this is the introduction. And what is cell? So we are all know it is an basic, fundamental and structural unit of all living organisms. So in NCERT we are going to see about what is a cell. So unicellular organisms are capable of independent existence and performing the essential function of life. Though in unicellular organisms we are going to see about the only one cell. So in cell there are two capabilities. What are all they? First one is independent existence and another one is performing essential function of life. So what is independent existence? What is independent existence? It does not depend on another cell, okay? Only one cell it is having, that is in unicellular organisms, that is only one cell is handling everything, that is respiration or breathing and then excretion like this and all, the only one cell, because overall only one cell is handling these are all the things, right? And it is performing an essential life. As we like that only one cell is performing everything, right? So here they are the best performing functioning material, the one cell, single cell, and anything than a complete structure of a cell does not ensure independent living. What is that? Anything less than capability, that is if the cell is having a structure, right? If cell is having a different types of structures, different functions, because one cell is having different types of cells will go for different types of structures, and hence cell is the fundamental structural, we are already seen that cell is an fundamental and structural unit of all living organisms, including unicellular and multicellular. So who discovered cell? Who discovered cell? So he discovered what? Anton von Leeuwenhoek is right. So what he discovered? Only one cell he is discovering. Either so if he discovered, he proposed a living cell or a dead cell. So he proposed a living cell. Anton von Leeuwenhoek is proposed living cell. But the first cell, dead cell, was proposed by whom? Dead cell was proposed by whom? Robert Hooke. He proposed a dead cell. Okay, and then Anton von Leeuwenhoek, he proposed a living cell. He invented a cell by seeing an live cell. He proposed. So in your NCERT, Anton von Leeuwenhoek was first and described a live cell. It is an MCQ. It is an NEET MCQs. So he first discovered, Anton von Leeuwenhoek was first discovered a living cell. Live cell. In living cell, he was discovered that it is an cell. And who discovered nucleus? Another point. Yeah. So Robert Brown later discovered the nucleus. In cell, it is having some nucleus, one nucleus. So we are having in prokaryotes, eukaryotes. So we are all going to see about what is prokaryotes, what is eukaryotes and all. So there are an nucleus, true nucleus, right? So in which in prokaryote or eukaryote, which is having an nucleus. So we are all going to see about that. So Robert Brown, Anton von Leeuwenhoek was first saw a living cell who discovered, and then Robert Brown was discovered nucleus. And then the invention of the microscope and its improvement leading to the electron microscope revealed all the structure details of the cell. So after that electron microscope came after 1900. Yeah. So after 1900, electron microscopes was invented, and then after that every single cell is having a structure and some functional units like that they are invented, they are proposed. And then what is cell theory? Here also we are going to see about some botanist, zoologist of who invented cell. Who invented cell in the sense what are all the things in cell theory we are going to see. So in cell theory in 1838 Matthias in this cell division, each and every point is important because it is having some years and all. 1838, 1865, who are the zoologists, who are the botanists. So they are asking like they will ask like that, who are the botanists in your NEET examination. So what is cell theory? In 1838 Matthias Kleeden, a German botanist, who Matthias Kleeden is a German botanist. 1838 he examined a large number of plants and observed that all plants are composed of different kinds of cells which forms the tissue of the plant. So in plant he discovered, who discovered? Matthias Kleeden in 1838 discovered that he discovered and he was examined through what? Plant cell. So every cell has, every tissue is having a cell. Like that he was examined, Matthias Kleeden. And then at about the same time, that is 1838, in 38, Theodor Swan, that is in 1839, after 1 year. So according to that, here Theodor is a zoologist. Then who is the botanist? Matthias Kleeden is a botanist. In 1838 he was examined a plant tissue, plant cell, plant tissue is having a cell. In 1839 Theodor Swan he's a British zoologist. He discovered some things. He studied different types of animal cells. So in Theodor was studied about different types of animal cells. In animal cells he was examined. In Matthias was examined in plant cells. Okay, and reported the cell had a thin outer layer which is today known as plasma membrane. Okay, so Theodor was examined in 1839 and he was examined in a animal cell, And what he was studied, what he was reported in the sense, he told that every cell is having an outer thin layer, right? Thin layer. Every cell is having a thin layer. That layer is called an plasma membrane. So now it is called an plasma membrane. In older days, outer thin layer only they are said. Okay. So plasma membrane and cell membrane, these two words are same. Either in NEET, your examination paper may also ask plasma membrane or cell membrane. Two is right. Okay. So here it is in the outer thin layer is called plasma membrane, that is cell membrane. He was discovered. Who discovered? Theodore Schwann in 1839. In plant cell, he was examined, he was prepared, and he was reported that the cell is having an outer layer. Then he also concluded. Who also concluded? Then he also concluded in your NCERT, he also concluded. Like that it is saying. So who also concluded? Theodore also concluded. So he also concluded based on his studies on plant tissue that the presence of cell wall is a unique character of the plant cell. So Theodore Schwann is proposed animal cell is having an outer thin layer. So and also he concluded that this study of plant tissue is also having a cell wall. So this animal cells and plant cell is having an unique character. So what are all the unique character? On the basis of this key one, that is Theodore Schwann proposed the hypothesis that the bodies of animals and plants are composed of cells and product of cells. That Theodore was concluded that animal cell and plant cell is having a cell. But and the product of cells also he was discovered. But only they have discovered cell is a unique function, that plant cell or animal cell. These two cells have a unique function they was proposed, but they don't know whether it is where the cell will grow. So they won't know, no, because in that time in 1838, 1839, no electronic microscope is not there in the 1838 and all, because after 1900 only we are having some microscope, electron microscope. So on that time they won't examine, right? So in that, after that, one scientist came. So before that we had seen about Schwann and Schwann together formulated the cell theory. This Schwann and who else? Matthias Clayton was, the both was formulated cell theory, because they both only discovered in plant cell they are having cells, and in animal cells also we are having some cells. They both only discovered, right? So followed by the theory, however, did not explain how the new cells were found. Even though there was found a cell, but they do not know how the new cell is occurring in our body. Okay. So after new cells, who discovered? Rudolf Virchow. Rudolf Virchow, 1855, he was discovered and explained that a cell is dividing. That is, cell is dividing. Cell is growing one by one, right? So we are having trillions of cells in our body. So cell is producing another cells, trillions of cells. The cell is dividing. So who discovered whether the cell is dividing? As in, so Rudolf Virchow was invented, proposed in 1855, that is, cell was dividing from the pre-existing cells. So what is pre-existing cells? What is pre-existing cells? The old cell. So here it is an old cell, this is an old cell. So in this old cell, one cell is developing, one cell is dividing. So we are going to see about the cell division in the third chapter. So cell is dividing. Who invented? Whether it is, the cell is dividing from the pre-existing cell. Who invented? Rudolf Virchow in 1855. So you have to remember the years and all. 1838. In 1838, who invented plant cell? Matthias Cleiden. And then in Theodore? 1839. 1838, 1839. So he invented animal cell is having a cell, the outer layer, the outer covering, right? And then they both discovered and cell theory. After that, Rudolf Virchow was invented what? The cell is dividing from the pre-existing cell. So who said? Rudolf Virchow in 1855. So you have to remember that year and all. It will be helpful for your examinations. Okay, then. So pre-existing cells, that is omnicellular or e-cellular. Omnicellular and e-cellular is an pre-existing cells, that is. So what is omnicellular and pre-cellular? Omnis, what is omnis? Omnis means new cell. New cell is producing by the pre-existing cell, that is old cell. Okay. Then he modified the hypothesis of Cleiden and Schwann to give a cell theory a final shape. Who gave a final shape to the cell theory? Because plant cell is having a cell, animal cell is having a cell. So both the scientists was invented, that is botanist and zoologist. So after that, Rudolf Virchow made an whole final shape to the cell, because Rudolf Virchow only invented the cell, whether the cell is dividing or not, he was discovered, right? Rudolf Virchow. Then cell theory, and today we are going to understood a cell theory in a two steps. What are all they? First one is all living organisms are composed of cells and product of cells. We are all know. So all living organisms, either plant cells, animal cells, humans. So everybody is having a cell. Every living organism is having a cell and having a products of cells. So what are all the products? Inside the cell, what are all the organelles? What are all the parts in the cell? What are all the parts in the cell? Yes, mitochondria, ribosomes, nucleus, cytoplasm. So these are all the parts. So we are knowing that the cell and product of the cell is very important to our organisms, one living organisms. The second, the cells arise from the pre-existing cells. We have known that. Who discovered pre-existing cells from a new cell? Rudolf Virchow. Okay, this is the 2 points we have to remember. Okay, and then after we are going to move into the overview of a cell. Can we? Do you all any doubt? Okay, fine. So an overview of cell. So an overview, what are all the overview we are going to see here? What are all the overview? What are all the overview? So we are going to see here is plant cell, animal cell. So what are all the shapes and structures, and what about eukaryotes? So because in eukaryotes means what? Presence of nucleus is eukaryotes. That prokaryotes? Absence of nucleus is an prokaryote. So we are going to see about, so in which plant cell is, what are all the organelles, that is parts present in the animal cells, what are all the parts present in the human cells, plant cells, we are going to see in the overview of a cell. So in eukaryotes. So eukaryotes is having what? Eu means what? Karyotes means what? U means true. So we are going to see about eukaryotes. So what is eu? What is karyotes? Eu means true. Karyotes means? Karyotes means nucleus. So we are going to see about the nucleus structure. So in eukaryotes, eu means true, karyotes means nucleus. So, true nucleus are going to see, and then nuclear membrane, nucleus, chromatin reticulum. So, these are all the things we are going to see, because it is having an inside the cell only these parts are there. So, in plant cell, animal cell we are going to see. Okay. You have early observed cell in an onion peel and or human cheek cells under the microscope. So, we are going to observe, just imagine we are going to observe our onion peel, that is an what? Onion peel, the onion is having some peels and all, no. So, we are going to see about the onion peels and the human cheeks. So, like a lump amount of human cheeks they want, just a small bit we can take for an examination. So, onion peel or human cheek we are going to see about see in the microscope under electron microscope, like, okay. Let us recollect their structures. So, we want to recollect the structures, because in one slide we are going to put the human cheeks or a bit of onion peels and all, including some solutions, because by adding some solution we can able to see more visibly, right. So, we are going to see the proper structure in the electron microscope. The onion cell, for example, an onion cell, it is an plant cell. Human cell is animal cell, right. So, here we are going to see about the plant cell and animal cell. Okay. Here we are going to see about an overview of a cell. So, plant cell and animal cell. In onion we are going to see. So, the onion cell, which is a typical plant cell. Onion cell is a plant cell, we all know that, and has a distinct cell wall as its outer boundary, and just within it is called a cell membrane. So, in onion cell it is an outer membrane, outer bond. So, it is having an cell membrane, that is plasma membrane. Cell membrane and plasma membrane is same. And then cell membrane and the cell of the human cheek. So, in human cheek we are going to see. So, it is an outer membrane and as an delimiting structure of the cell. So, it is an delimiting structure. So, it is having an double layered, and this is having because plant cell is having cell wall, animal cell is not having cell wall, right. So, here it is absent, here it is present in animal, because in both animal cells and plant cell you have to differentiate each other, because you may ask some questions in your NEET examinations. And then inside each cell is a dense membrane bound structure called nucleus. So, inside plant cell and animal cell it is there is a nucleus, right. So, this is a nucleus. So, here, so inside the nucleus we are having what inside the nucleus? What? genetic material we are having chromosomes and all, genetic material in the sense DNA, RNA we are having in the nucleus, inside the nucleus. So, and then this nucleus contains a chromosomes. So, you have just now you have told that nucleus, it is having an inside the nucleus, they are inside the nucleus some genetic materials are there. So, in your NCERT it is telling that this nucleus contains some chromosomes in which in turn contains a genetic material DNA. So, here chromosomes, so genetic material is containing like this and it is an dense, right, it is an dense, sorry. So, inside the nucleus we are having a genetic material, that is chromosomes, DNA, cell that have membrane bound nucleus called eukaryotes. Membrane bound nucleus. What is, who is having membrane bound nucleus? Eukaryotes. Animal cell is an eukaryotic cell, right. So, eukaryotic cell is having a membrane bound nucleus. Membrane bound nucleus. And then whereas cell that lack a membrane bound nucleus are prokaryotes. It is the animal cell is having membrane bound nucleus, whereas plant cell is having what? Here animal cell is having animal cell will not have cell wall, plant cell is having cell wall. So, animal cell is a membrane bound nucleus. So, that is true nucleus, whereas plant cell lack of membrane bound, So, you have to remember, okay, lack of membrane bound. So, lack of membrane bound in prokaryotes, this is prokaryotes. And this is eukaryotes. This is true nucleus, this is absence of nucleus. Okay. Then, in both prokaryotes and eukaryotes, a semi-fluid matrix called cytoplasm occupies a volume of the cell. So, in plant cell and in animal cell, that is eukaryotes and prokaryotes, these two cells containing an what? Containing what? A semi-fluid matrix that is an cytoplasm. So, it is the both is having an cytoplasm. So, the both cytoplasm is having an semi-matrix fluid. What is semi-matrix? Semi-fluid. Semi-fluid matrix means semi-fluid. So, liquid, say like semi-liquid. Semi-liquid. For example, what is semi-liquid? Jelly type, that is solid. So, vazha irukum appdi solvaanga illaya. So, that is an semi-fluid matrix. So, that is an cytoplasm. So, that cytoplasm is having an cell volume is having a cytoplasm. And then the cytoplasm is the main area of cellular activities. What is area? What is area? What is area? Like a ground, playground. No, we are playing in the playground like that. Cytoplasm occupies the cytoplasm is the main ground for cellular activities. Some activities is doing from the cells. So, the cytoplasm will occupies the main area for activities of both plants and animal cells. Various chemical reactions may occur in it to keep the cell in the living state. So, in cytoplasm only, because cytoplasm is having a semi-fluid matrix, right. So, that is on a big ground like that is area. So, for cellular activities, so it is both for both plant cell and animal cell for chemical reactions. Chemical reactions means what? What are all the chemical reactions? What are all the chemical reactions? have already seen, right? So cytoplasm, so that is having some chemical reactions. So in some chemical reaction will be taking place in the cytoplasm. So if that reactions will take place, the cell will be in the living state. That the statement is given here. And besides the nucleus, the eukaryotic cell have other membrane bound distinct structures called organelles. So beside the nucleus, beside the nucleus, what are all the things having means, so beside the nucleus in eukaryotic cell, that is an membrane bound, we know that eukaryotes having an membrane bound nucleus. So in membrane bound nucleus, what are all the organelles? What is organelles first? Distinct structures of, it is an membrane bound nucleus, so it is having some organelles. What is mean by organelles? What is mean by organelles? What is organelles? Organelles means what? What? That is organelles, huh? What? So organelles is nothing but organs present inside the cell. That is organelles. Organelles means what? Organelles. Organs present inside the cell. So what are all the organs? What are all the organs? Endoplasmic reticulum, Golgi complex, mitochondria, chloroplast. So these are all the organelles present inside the cell. So parts, that is parts, organs in the sense parts present inside the cells. Then, so what are all the parts? Endoplasmic reticulum, Golgi complex, lysosomes, mitochondria, and microbodies and vacuoles. So these are all the parts present inside the eukaryotic cells. So we are going to see one by one, what is endoplasmic reticulum, what is Golgi complex, what is lysosomes, we are going to see later on. Okay. Then in prokaryotic cells, we know that prokaryotes is having a lack of membrane bound nucleus. So in prokaryotic cells, such membrane bound organelles will be lack, because here cell wall present, here cell membrane is cell wall is absent, cytoplasm. So like that some other organelles will be present here, or some other organs may be absent here. So we are going to see on later. And then ribosomes are non-membrane bound organelles. So here lack of membrane, here membrane bound organelles. So non-membrane bound organelle, ribosomes. What is ribosomes? Ribosome is an non-membrane bound organelles. So what is what is ribosomes? Protein. So synthesis of protein. So here we are having some ribosomes and all. So in ribosomes we are having in endoplasmic reticulum. Endoplasmic reticulum will be split into two types, that is smooth endoplasmic reticulum and rough endoplasmic reticulum. So here we are having some protein synthesis, that is ribosomes, sugar. So ribose sugar. So ribose sugar means here we are having some sugar. So that is ribosomes, that is protein is synthesizing. Where protein is synthesizing? From where? You are telling right, ribosomes is protein synthesis of protein, sugar. So where protein is synthesizing from? RNA. Okay. Ribosomes are non-membrane bound organelles found in all cells in the sense both prokaryotic as well as eukaryotic, and ribosomes is present in prokaryotes and eukaryotes. Ribosomes, here also ribosomes is present. And then Within the cell, ribosomes are found not only in the cytoplasm but also within the two organelles. So, ribosomes is present in the cytoplasm but not only in the cytoplasm, it will be seen in other two materials also. So, what are they? Chloroplast in plants and mitochondria in rough endoplasmic reticulum. So, in plant cell it is having a chloroplast, animal cell it is absent, right, because it gives in colors chloroplast. So, in plant cell this ribosomes are present in the chloroplast, and then cytoplasm mitochondria and on rough endoplasmic reticulum. Here you are seeing this is an just imagine this is an endoplasmic reticulum. So, we are having some ribosomes here. So, that is present in the rough endoplasmic reticulum. In rough endoplasmic reticulum only ribosomes will be present. Smooth endoplasmic reticulum, there won't be present in the ribosomes. Next, animal cell contains another non-membrane bound organelles called centrosome, which helps in cell division. So, in first we have seen that cell was dividing from the pre-existing cell. Who proposed that cell was dividing by the pre-existing cells? In which year? 1855. Rudolf Virchow was proposed that cell is dividing from the pre-existing cells. So, here in animal cells in animal cells also on other non-membrane bound organelles called centrosome, that is in the cell division. Centrosome is helping. Centrosome will help while dividing the cell. Centrosome is helping. Okay. And then cells differ greatly in size, shape and activities. So, cell already I have seen in the first cell. So, cell was divided greatly differs from shape. One cell will be in different shape, another cell. In human cells we are having red blood cells, erythrocytes. But in white blood cells we are having different types of cells, that is leukocyte, erythrocyte, basophils, lymphocytes. So, like that and all we are having different types, different shapes in cells. So, we are having some size, shapes and activities. For example, mycoplasma, the smallest cell. So, the smallest cell, here the NEET question. So, which is the smallest cell? Mycoplasma. And what is the length? 0.3 mu meter in length. So, here we are seeing that smallest cell plasma and its length is 3 mu meter in length. And next we are going to see which bacteria could be 3 to 5 mu meter. The largest isolated single cell is called egg, an ostrich egg. So, the largest, this is an smallest cell. The largest single cell, which is the largest single cell? Largest single cell, egg of the ostrich. Okay. Sorry. Among multicellular organisms, human red blood cells are about 7.0 mu meter in diameter. So, in human, this is all single cell organisms. So, in human cells, we are having different shapes, size and all. So, we are having how much? Red blood cells, 7.0 mu meter in diameter. So, diameter is in human cells 7.0 mu meter in diameter. This is in length. This is in length. And then nerve cells are some of the longest cell. What is? nerve cell. So you are seeing in your NCERT pictures, that is diagram in 8.2. So you are seeing the nerve cells. It is like what? Yes. Sit down. So in nerve cells we are having, some nerve cells, like, see, this is a nerve cells. So here we are having a branched of nerve cells. In your NCERT book, you will see the pictures in figure 8.1, you will see. So here the branches of nerve cells. So this is the longest cell. Which is the longest cell? Nerve cell is the longest cell, and cells also vary greatly in their shape. So what are all the shapes cell will differ from? What are all the shapes? Tell me. What are all the shapes will, because after next point is that only. So what are all the shapes? So we'll see. Okay. Okay. Cell also vary greatly in their shapes. So what are all the shapes you are going to see? They may be disc-like. What is disc-like? What is disc-like? What is disc-like? What is disc-like? Circle. Are the RBC, erythrocytes, are erythrocytes. So that is an erythrocytes. So here disc-like structure. You will see in your NCERT figure 8.1, our RBC will be here, right? You are seeing, right? So that is a disc shape. And then what is polygonal? What is polygonal? Our cell, we seen about our overview of cell, no? This is polygonal shape. And then columnar. What is columnar? Columnar. Column shape. And then columns in your NCERT, column shape, this. Okay. Sorry, this is row. This is column shape. And then next, cuboidal shape. What is cuboidal shape? What is cuboidal shape? Like cubes. Like this. Okay, cuboidal shape. And then thread-like or even irregular, thread-like structures. Thread-like, like thread-like. So this is an irregular structure. What is irregular cell? Give some example for irregular cell, irregular structures. Amoeba, because it does not having an structure, same structure. So amoeba is having an irregular structure. So the shape of the cell may vary within the function they perform. So every cell is having some functions and nature and all. So every cell is having some functions, some enough, no need. So every single cell is having some functions and they perform it through the cells, because a red blood cell is having a disc-like shape. So it is producing some hemoglobin to our body, and then in cuboidal, some other in nerve cells. So each and every cell is different from their single size and shapes. So it is functioning like that also, unique character. One cell is having an unique character. As we have seen that in prokaryotes and eukaryotes, the cells is having an unique character in eukaryotes and prokaryote cells. So, and then we are going to see about the prokaryotic cells. So prokaryotic cells we will continue by tomorrow. Okay. So tomorrow at this time we will see